then section, again, that section yet this week, download and reprint number 4 and look at it. It has to do with RNA synthesis in rhabdoviruses, which would be like BSD and rabies. Supplementary readings, chapter 15. As a reminder again, our first midterm is Monday, and if your last name begins A through K, or you're in the DSP program, come to this room. If it's L through Z, go to Buchanan, 1940. And there's a practice exam on the website to give you a sense of the kinds of questions, style of questions I typically ask. I'll have a question and answer session today here in this room at 4 o'clock. Answer any questions you have about virology. One question I did get from a couple of people on Wednesday relates to section, and does the midterm cover section? And the answer, I guess, is yes, in the sense that the papers in section were picked to coordinate with the information that we're considering in the lecture, and the section should have given you an experimental sense of what the evidence is for some of the topics that we consider in the lecture, like the first week, the plaque assay, second week, receptors and tropism with polio, and then this week, of course, transcription, and then we touched upon the UV inactivation. So, all right, any questions before we begin today's discussion? Yeah.
Cover up the VSV or VSV?
Good question, because I think I mentioned on Wednesday, the midterm will cover up to and including rhabdoviruses, but the midterm will not cover myxoviruses, which would be measles, that kind of. Okay. Because the myxoviruses will be a couple of lectures. Not measles, but BSB. Any other questions? Hopefully that's all right, where we left off, can you close the doors so the light is a little better in the street? Where we left off in our discussion of BSV is we had receptor-mediated endocytosis and uncoating removal of the envelope, which activated the polymerase. The polymerase is virion associated, and the polymerase is acting in the transcription mode. So we don't need protein synthesis. Simply removal of the envelope activated the polymerase. We're in transcription mode, so we're making the mRNAs. So from this one large single-stranded negative sense molecule, that was the parental genome, we make 5 different mRNAs. And so the process by which this happens, and this is figure 3 of your handout, is there's a single entry site for the polymerase at the 3' end of our minus genome, and the polymerase initiates translation. We make a short leader, and then we first transcribe the N gene. And so the polymerase then will come to what we call an intergenic region. Intergenic, simply a sequence between the genes. So there's a sequence between the N and P, the P and M and G, etc. So this intergenic sequence. And in the transcription mode of these viruses, and you have this figure on your handout too, but the orientation now, keep in mind, is opposite, 3' to the right. So we initiated the polymerase, we transcribe through L, we initiate, we transcribe through L, then we're in the N gene, making the N messenger RNA, and we come to this intergenic region, and that's the white region on that schematic on your handout. And there are two nucleotides that are not transcribed. So what happens, so in the intergenic region, the actual sequence, so we start, it's our minus single-stranded RNA genome, 3' end, the Pol entered, and we transcribe, and it comes to a sequence in the minus sense, in our template, and the sequence at the end of the N gene is AU, AC, and then there are 7 U's, all right? And then there's a dinucleotide, GA, dinucleotide, those 2 nucleotides. So in the intergenic regions, there's these 2 nucleotides in each of the intergenic regions that will not be transcribed. They're not transcribed. Then we come to the next intergenic sequence that happens to be U, U, G, U, C, then blah, blah, blah. We're in the P gene now. So what I wrote for you is the end of the N, the intergenic sequence, and what will be the 5' end of the P. This would be the N gene, P. All right, so what happens with our polymerase now, the virion-associated polymerase, we're making the N message, so the polymerase is going down this minus single-stranded RNA, and now we're at the what will be at the, what will be the end of the 3' of the N message, because the complement to this, of course, is 5'. This is the 5' end of the N message. We're making N message, so it would be the complement of that sequence, U, A, U, G. And then the polymerase hits these 7 U's, and it sputters. It just keeps putting in A's. It slips, and it puts in the order of 75 to 150 A's, templated by those 7 U's. And that's how you make the 3' poly A. So the messenger RNAs are 3' polyadenylated, but you get that poly A by virtue of the slippage that occurs when the polymerase hits the 7 U's. And then what happens is that dinucleotide is not transcribed, but a certain fraction of the polymerases don't leave the template. This is our template. They don't leave the template, but then they start synthesis at the subsequent sequence, which would be A, C, A, G, this is a C, etc. So this becomes the 5' end of the P mRNA. So this sequence at the 3' end of the N, prior to the U, and the sequence following the GA are conserved at all the junctions, as are the dinucleotides, with one exception. So not all of the polymerases make it through that dinucleotide. And that's what we mean when we see those attenuation that occurs. The polymerases that are transcribed started at the very 3' end of our template to make N message. That's the only entry site on the template, on the genome. So we make N, we stutter, and then some of the polymerases don't leave the template and make it through that dinucleotide and start transcription of the next gene, that's P, but you lose some polymerases, and that's the attenuation. So this happens at each of the junctions. So the consequence of that is the attenuation is the basis of regulation. And so of the mRNAs that are produced, and that's why these lines in my schematic that I made for you, the cell, the N is pretty thick and the L is much thinner. center, the molar amounts, you make way more N message than you do P, than you do M, than you do G, than you do L. Because at each of the junctions, and S was P, remember that? So each of the junctions, you lose some of the polymerase. So by the time you get down to transcribing the L gene to make that mRNA, you've lost polymerase at each of those junctions. So the molar amount is very high for N and very low for L messenger RNA. And then again, the U7 is what signals the polyadenylation. All right, so you have this progressive decrease in molar amount of messenger RNA made as you transcribe R minus single-stranded RNA genome to make these 5 mRNAs. And again, that transcriptional process is independent of protein synthesis. You do not need protein synthesis in the infected cell to transcribe because you brought the polymerase in with the particle. However, to switch to the replication mode. So in replication now, what we're going to do is make a full-length plus strand as opposed to the 5 individual plus strands, which were the mRNAs. We need to make that full-length plus strand because that's going to be the template to make full-length minus strand product that's going to be the progeny genome. So we switch then from the transcription mode to the replication mode. The minus single-stranded RNA is not being used to make mRNAs, but the minus is used to make a full-length plus RNA. So in the replication mode, that is dependent upon protein synthesis. So replication, it's protein synthesis dependent, and a protein that you have to make is N protein. Because N protein then goes back and in part acts as an anti-discriminator at the intergenic junction to allow you to make that full-length plus strand. The other thing that happens in transcription mode, so when we're transcribing and making those 5 mRNAs, in terms of the characteristics of the mRNAs, so our mRNAs that we made, the 5 different ones, as I just told you, at the 3' end, they're polyadenylated. So the mRNAs are polyadenylated because of that stuttering, slipping process of the polymerase that they use in the intergenic junction. So that's typical of a message in an animal cell, in a eukaryotic cell. The other thing that occurs to the mRNA is at the 5' end, the mRNAs are capped. And on your handout, you have this figure on the left, which is a cap structure that you may have learned about genetics, because all eukaryotic cellular mRNAs are capped. But this cap structure was discovered with viruses. It was discovered with BSE and reovirus. And so the addition of the cap is generated by an activity that's within the L protein. So the virus L protein has the activity to put the cap on for the viral message. In the cell, which I'm not going to go into, the cellular capping, just to review what you learned in genetics, happens within the nucleus as an early part of with transcription by RNA polymerase II, which of course makes message in an animal cell. But in the viral situation, all of this is in the cytoplasm. So it's an activity of the L protein that adds the cap. And so what the cap is, when you start transcription, of course, the 5' end will be a triphosphate, and then whatever that nucleotide is, and it's almost always a period to start transcription. So what happens with the cap is you end up post-transcriptionally adding a G. So the G that's added at the 5' end, the G that's added, that's what we call the cap. And it's a highly unusual bond in biochemistry, in that the bond is a 5' phosphoester linkage. That's a very stable bond. There are very few enzymes that can break that bond. And so it's protecting the end of the message. And then the other thing that happens is there's a methylation that occurs on the G at the 7th position of the period, which is depicted here. And so 7-methyl-G is the cap on the mRNA. And I want to keep emphasizing that. Like in polio, to go back a week and a half, polio turned off cellular protein synthesis because cellular protein synthesis is by a cap-dependent translation. And polio degraded a factor you need to initiate translation of cap mRNA. Well, it turns out that other viral RNAs like VSV and other viruses other than polio that replicate in the cytoplasm also have a cap like this structure. So, hey, what if I asked you a question? If you doubly infected a cell with VSV and polio, what would polio do to VSV protein synthesis? Inhibit, right? Because VSV needs the cap binding protein to initiate translation. Anyway, so these 5 mRNAs that we make in the rhabdovirus infected cell are 5' capped and 3' polyadenylated. And the 5' cap and the 3' poly A are made by viral enzymatic activity, slippage to put the A's on, and then the guanylyl transferase and methylation is also an activity within the L protein. All right. So in our multiplication scheme then, we've transcribed, to go back a bit here, we've transcribed, we've had attenuation, and so we make varying amounts of mRNA. The next step then after transcription is protein synthesis. And we make 5 proteins, N, P, M, L, and G. And the regulation of protein synthesis, the control largely goes back to transcription, because the amount of the protein that you make is determined by the concentration of the mRNA, and you had different concentrations of the mRNAs because of the attenuation that happened at the intergenic junctions, okay? So the control in terms of the amount of the different proteins we're making is dictated by how much mRNA there is for each of the proteins. And so among the proteins we make are the N protein. So that once we have the N protein made, then we can go back and activate RNA synthesis. And as I just said, we're in the replication mode as opposed to the transcription mode now. And this then allowed us to make the full length plus, which is allowing us to make full length minus by the L protein, which is the replicase. So in replication, L is functioning as the polymerase, but we need N that interacts, that prevents the termination and making the messages, and we also need the P protein. All right, so we replicate We've made proteins, and this allows us to make progeny nucleocapsids. And so here's another concept that we're going to introduce, in that we had the parental genome, the minus single-stranded RNA genome, the parental input, and this was transcribed to make mRNA that was translated to make the 5 different proteins. This expression off the parental input genome, we call this primary expression, because it's gene expression off of the genome that came in the initial infected particle. Once we've made the protein, like M protein, then we can go into we can go from transcription to replication, and in replication, then we're making the full length plus initially, that then becomes the template, so we can make full length minus. And that full length minus RNA that we make will become the progeny genome. So it's going to be encapsulated ultimately when we assemble our particles. But the other thing that can happen is it can serve as a template, additional template to make more plus, to make more minus. And so it can be a template in itself in replication, or we can make progeny nucleocapsid right here, and then these progeny nucleocapsids will have the L polymerase, and so this then essentially transcribes to make more mRNA and more protein. So we're transcribing off of progeny nucleocapsids. And so that expression is called secondary gene expression, because we're in it, and I've tried to depict it right here for you. Secondary transcription is going to feed in and make more of that message that's going to make protein. But this transcription is coming off of templates that we really synthesize, as opposed to templates that came in with the protein. In the infected cell, if you quantitate how much expression is primary off of parental genome and how much is secondary, that off of progeny, it would be expressing here. The vast majority is off progeny. So like 99% is coming here, in the order of 1% or so here. So you've essentially set up this factory, if you will, of making viral proteins. Does that make sense to you? In the regulation then, again, in order to have that occur by replication, we needed to make the N protein that prevented anti-termination, and then the P protein, which used to be called NS, but the reason it then got changed to P is because it gets modified with a phosphate. The P protein is a phosphoprotein. And the phosphorylation status seems to change, in that when we're in the transcription mode, in transcription, the P protein is phosphorylated. But when the polymerase is in the replication mode, when P is working with L, it's dephosphorylated. That is, it's not phosphorylated. So phosphorylation status of P affects its activity. Okay, so we've had this amplification template. We've built up proteins in our infected VSV or rabies cell. we've made progeny nucleocapsids. So how does so the next step is assembly of our progeny virions. And for these viruses, we say assembly is coincident with release. That is, the progeny particle isn't really assembled until it's released from the infected cell. And that release occurs by budding from the cytoplasmic membrane of the infected cell. So when the virus buds, what happens is that the nucleocapsid then acquires the envelope membrane of the particle, which is the case for rhabdoviruses. It's going to be the case for our next family, the myxoviruses as well. So, in terms of the polarity of the cells in culture, when this happens, this budding process has polarity in that the basolateral surface is more efficient than the apical surface of the cells where that budding is occurring in culture. And so, in budding, then, we acquire the envelope. And so the constituents of the envelope, the protein constituent of the envelope of our final particle, the protein components are viral gene products. And so this will be the G protein that's going to be modified to contain sugars and hence become the glycoprotein G protein. That's a viral gene product. The M protein is sometimes viewed as an envelope protein, the matrix protein. That's a viral gene product. But the other constituents, the phospholipid and cholesterol, i.e. the lipid components that make up the envelope, those components are silent because they're coming from the cell's plasma membrane. And they vary, depending upon the type of cell that the virus replicates in. Lipid constituent of the envelope is derived from the cell. Okay, so what's the mechanism? The G protein is viewed to be an integral membrane protein. And I'm going to return to this concept in a second because a lot of what we know about glycoproteins and cell biology came from the study of VSBG. It's an integral membrane protein because it's going to be synthesized and trafficked, that we're going to return to deliver the G protein to the plasma membrane of the cell. So the G protein is made, it's inserted into the plasma membrane of the cell with carbohydrate modification, the sugar modification. That's the whiskers on the diagram. After the G protein is inserted, then the M protein, so the N, P, M, and L, and the font size reflects the amount relative of the protein. So at any rate, the M protein interacts with the plasma membrane in the areas where the G protein has been inserted. So you have these clusters on the surface of the cell. And then, when this gets formed in the infected cell, then the nucleocapsids that have been transcribing or replicated, then they associate with the MG complex, and that triggers the budding. So they end up having a cell that's just budding off these particles with the envelope. In large amounts, one particle in might give 5,000 progeny out. So it's a very efficient process. again, the efficiency in terms of release in cell culture is basolateral, it's more efficient than bigger. Okay, so how about this G protein? What have we learned about glycoproteins from the study of the VSV G protein? That's an integral membrane protein. So these kind of proteins have 4 domains that were first worked out with the G protein. And so the 4 domains, the 5' end of the G gene ends up making the N-terminal end of our G protein, and this will be the C-terminal end of the G protein. So the G protein, the very 5' sequence, the G protein, I should say, is 511 amino acids long. The very N-terminal part of the protein, 16 amino acids. That domain is called the signal sequence. It's very hydrophobic, and it directs, when the G protein is first being made, it directs that nascent protein to interact with the Virology machine. So it directs, it's a hydrophobic sequence, very short sequence, directs interaction with the endoplasmic reticulum, ER, lumen. And then what happens, that very short sequence is going to get cleaved off. So then it gets, the sequence then gets cleaved off, removed by a signal, what's called a signal peptidase. So the sequence gets cleaved off, the N-terminus, once it's been used to direct into the ER bulges. The next domain of the G protein was large. In the case of VSB, it's 446 amino acids. This is called the body of the protein. It's the bulk of the protein, and it's going to be exterior to the envelope and plasma membrane. And this is the region that contains the sites to which the carbohydrates, i.e. the sugars, get added to make it a glycoprotein and modify. The spike projections, the glycosylation, occurs on the body. And the sequence to which that happens, and in a very common type of glycosylation, is called asparagine linked glycosylation. Because there's a sequence in the protein, it's asparagine, some amino acid X, serine, or C, and it's to that asparagine then that the carbohydrate gets added. The carbohydrate addition is initially as mannose, which is a sugar. Lots of mannoses get added, and then they get cleaved back, and then other sugars get added depending upon the property of the cell. Sugars like galactose, glucose, and sialic acid. Sialic acid is going to be big. We're going to return to that when we get to influenza. Anyway, so you add sugars and trim and add, and that gives the unique carbohydrate pattern, which we call the glycosylation. Sugars in the body. Then there's a domain, the third domain is called the TM domain, which is transmembrane domain. And that transmembrane domain is about 20 amino acids. And it's a hydrophobic sequence, i.e. amino acids that are uncharged. And that's the domain that spans the lipid bilayer in the membrane, transmembrane. And then the C terminus 29 amino acids is called the cytoplasmic tail. And effectively, it's an anchor. Usually highly charged amino acids. So the amino acids have charge, and that anchors the glycoprotein in the membrane. If recombinant DNA technology is used to express the protein without that tail, it just shoots right through. It's not anchored. So the ultimate orientation, which is a common orientation for glycoproteins, the N-terminus ends up outside, and the C-terminus ends up inside of the cell, which is depicted by the N and C. And so this was worked out from the study of VSB, glycoprotein G, and it's true for the vast majority of viral glycoproteins that we will consider, including the main ones of HIV, that are a hot topic these days. All right, so that's how we made our glycoprotein and how we get our project viruses. What's the virus doing to the host cell during this replication? So what's happening to cell metabolism? All right, so one thing that happens is we have an inhibition of cellular protein synthesis. And so the inhibition seems to be mostly by a competition mechanism. Because the replication in this case is so efficient that the concentration of mRNA that's viral is so high relative to the concentration of any given cellular RNA. So the viral RNA just competes out the cell mRNA and ends up shutting off translation. And then there's an inhibition of DNA-dependent transcription, i.e. cellular transcription. And this is believed to occur by the short reader RNA that was made, that the viral RNA gets targeted to the nucleus, and that's what's depicted on the schematic there, and leads to an inhibition of cellular transcription by the leader or L viral RNA. Inhibits Paul 2. Okay, so that's how a rhabdovirus replicates. We learned a lot from the study of VSB. Now let's switch for a bit to rabies, because rabies, while VSB is important in ag in terms of cattle and horses, rabies is medically important. Human can be important. So what about rabies? So rabies as a disease is caused by rabies virus. It's a rhabdovirus. The replication scheme is very similar to what we just discussed with VSB. Rabies is a significant global disease. Estimates are there are about 30,000 deaths per year from rabies, mostly in Africa and Asia. Very, very rare in the U.S. Human rabies cases. Very rare, as you'll see in a second. Rabies is what we call a zoonotic disease. Zoonotic because it's transferred to the human from an animal reservoir. Human-human transmission is exceedingly rare. The last example that I'm aware of was from an organ transplant where the organs came from the individual that happened to have rabies. Very sad situation. Human-human is exceedingly rare. Zoonotic, animal-human transmission. And so the transmission is via saliva in a bite by an animal to transmit from the infected dog. Outside of the U.S., globally, So the domestic dog is the usual vector source. In the U.S., dogs and cats are immunized, so we rarely see the disease. But there are numerous reservoirs. And that's depicted here. You do not have this on your handout, but it's showing epidemiologic data from 2001 to 12, and then I gave you the latest data for last year that I could get. This is rabies in California. And so in California, it's mostly bats, which is the blue bar, and skunks, which is the red bar. Typically about 200 to 250 cases per year in California in animals, not humans, animals. And there 150 plus in bats, skunks, much less, 10 to 40, depends on the year. And then, the next I would say, so bats, say 150 plus, skunks 40 to 40, and then other, and the other might be a case or two of an occasional cat, dog, coyote, are the main ones. And last year, 183 bats, 22 skunks, 2 cats, 1 coyote, no foxes, and no dogs. How about the human? So, in the period 2001 to 2015, there were 8 cases of rabies. And as far as I'm aware, the last fatal case was actually in Santa Barbara County, in Santa Maria. And the individual had been affected, and this illustrates another point. Of the 6 cases I'm showing here, 4 of the 6, the infection occurred out of the country, in the individual country. And that could be proven by isolation and sequence of biopsy. And, the vector was dog or bat in these cases. But, the main point is human rabies is exceedingly rare cases in the state and in the country. All right, so how does the disease work? Why is a rabies infection, if it occurs in the human, frequently fatal? So the pathogenesis, in terms of progression, it's what we call a neurotropic disease. So initial inoculation is from the saliva and the bite, bitten by a dog or a skunk or whatever that has been infected. You have replication of the virus through the scheme that we've dissected with VSV, molecularly, similar scheme for rabies. Replication at the bite site, then spread as you increase the amount of virus to the central nervous system. And this occurs quickly, the virus replicates quickly, so it happens before there's any significant immune response. In the central nervous system and peripheral nerves, virus enters, replicates, spreads, spinal cord, ganglion, spreads to the brain, the ensuing encephalitis. And the progression is rapid. It's because of that rapid amplification and progression that then typically coma, that point on rhabdoviruses and rabies. And now let's switch gear. Are there any questions on rhabdo? Okay, let's switch gears now to our next virus family. And there's actually two families that I'll just briefly introduce, and then we'll stop on time. And these are negative strand viruses, and this will not be on good, right? Because they're mixoviruses. And so where we're going to be, we just finished rhabdoviruses where the genome's negative, we have serine associated polymerase that gives our message, and then the cell machinery can make protein. The same is true for mixoviruses. So, but with mixoviruses, just to give you the overview, mixoviruses, Viridae virus family, Mixomucus was the current host. Because the viruses bind peptides that are mucopeptides that have the modifications. So they bind mucopeptides is where the name came from. They glutinate red blood cells. Hemagglutination is a common acid. So of the mixoviruses, we will have paramyxoviruses and we will have orthomyxoviruses. And they're both negative strand. They have minus single-stranded RNA as the genome. But it's going to be really different in the sense that paramyxoviruses have one species, so they're monopartin. And our prototype virus here is going to be measles, which is an important disease virus globally, yeah. After the Disneyland situation a couple of years ago, a lot changed in California this year. We'll see it less and less of an issue. But measles disease caused by measles virus globally is still a huge problem. So that will be our prototype for paramyxoviruses. Orthomyxoviruses that have a minus single-stranded genome, this is a new concept for you. There will be 8 different molecules, 8 different minus single-stranded RNAs. So these are viruses that we call segmented, because the genome is segmented. And as a consequence of having a segmented genome, the frequency of genetic reassortment is much higher, because you have 8 different pieces of RNA. So if you infect with 2 different viruses, you could reassort those pieces of RNA. Our prototype virus here will be influenza. This is why influenza is still one of the major threats to mankind, because you get genetic reassortance and generation of a brand new virus to circumvent immunity, which happened in 1918, and as a consequence, more people died from a flu infection than died in the war. So, I probably owe you 2 minutes. We'll stop 2 minutes early, and then after